Good morning, ladies and gentlemen. The magic hour is at hand, and I think we should probably start. For those who may not know me or who do not know me, my name is John Thompson. I'm Professor Emeritus in Biology, and I'm also Senior Associate Vice President Research here at the University of Waterloo. And it's a particular pleasure for me to welcome you, on behalf of our President, Feridun Hamdalapur, and also the Vice President of Research, Charmaine Dean, neither of whom are here today. Sometimes Feridun manages to drop in partway through, and he may or may not be able to do that today. Last I heard he was in Switzerland. I'm not sure if he's back yet or not. So a little bit about UW Research Talks. They are jointly sponsored by the President and by the Vice President of Research, and they are meant to provide an opportunity for all members of the university community, students, staff, and faculty, to learn firsthand about the important and exciting research being conducted right here at the University of Waterloo, where we all work. And today's session is in the form of a panel discussion, and we have 4 exceptional panelists who agreed to participate, and they're still here, who will start the session by sharing with us some of their thoughts on the topic. And the topic is a very interesting one. It is unearthing ancient environments with microbes. So I think we're in for a good time. Following these brief presentations by the panelists, and the last I heard you had 5 to 7 minutes, something of that order. Following these brief presentations, our panelists will then facilitate an open discussion on the topic with you people, members of the audience. So Alexis Dolphin will be first off the mark. Alexis, can you identify yourself, please? Thank you. Alexis is an assistant professor in anthropology and a member of the Waterloo Center for Microbial Research. That's an interesting combination, I must say. She specializes in dental anthropology, human osteology, bioarchaeology, paleonutrition, and paleopollution, with a particular emphasis, as I understand it, on reconstructing early environments to better understand infant and child experiences. She has conducted field work, and I'm envious of this, and analyzed samples in Ontario, well, that's okay, Belize, Iceland, Egypt, Mexico, Norway, and Sudan. That's amazing. Alexis holds a PhD in anthropology from the University of Massachusetts, a master's degree in anthropology with a specialization in bioarchaeology from Western, and a bachelor's degree in anthropology from McMaster. Prior to her professorial appointment here at Waterloo, she completed a postdoctoral research fellowship at Western and was a full-time faculty member at Western before coming to Waterloo. Thank you, Alexis, for agreeing to be part of this discussion today. Next up will be Andrew Doxey. Andrew, if you could identify yourself, please. Andrew is an assistant professor in biology. He's cross-appointed, though, as well, to the Department of Computer Science, and he also holds an adjunct professorial appointment in the Department of Medicine at McMaster University. He leads the Applied Bioinformatics Group, is a member of the Waterloo Center for Microbial Research, and the Center for Bioengineering and Biotechnology. Andrew, I think he describes himself as a bioinformatician. Have I got that right? With research interests in comparative genomics, protein function prediction, which is a very important area, and molecular evolution. He and his team use computational methods to discover new genes and to elucidate protein function. Andrew holds a PhD in bioinformatics from Waterloo. Congratulations. And he completed an NSERC postdoctoral fellowship at Stanford University before returning to Waterloo in 2013 as a full-time faculty member. We're really pleased that we got you back, Andrew. He has received a number of awards, most recently the Thermo Fisher Award from the Canadian Society of Microbiologists, the Science Excellence in Early Career Research Award, and Waterloo's Outstanding Performance Award. He has published over 40 peer-reviewed articles, which at your stage of career development is very significant. And this is very important. He recently discovered a new Botox protein in collaboration with researchers from Harvard. And you're going to tell the group what a Botox protein is, right? Okay. Thank you, Andrew, for participating today. We really appreciate it. Next up will be Kirsten Mueller. Kirsten is a professor in the Department of Biology and also holds an administrative appointment. I don't know how you managed to get roped into this, Kirsten, as the president's not here, right? So as Assistant Vice President of Graduate Studies and Postdoctoral Affairs. She's also a member of the Water Institute and the Waterloo Center for Microbial Research. Kirsten is considered to be a world expert on Bangiales. Now, I'm not sure if I'm saying that correctly. Bangiales, okay, thank you. An order of red algae that is an ancient lineage with some members in the fossil record going back as far as 1.2 billion years. In addition to their significant economic importance in food and aquaculture, red algae are a critical group in the evolution of photosynthesis. Very little is known about this group, including their apparent, I think it is apparent, inability to undergo sexual reproduction. It wasn't clear to me. And Kirsten's research focuses on speciation, taxonomy, and evolution of sexuality within this enigmatic group. She and her team use molecular techniques to discern if sexual reproduction is or is not occurring within populations. Kirsten received a PhD in botany from the University of Guelph. She currently serves as president of the Phycological Society of America and directs the Canadian Phycological Culture Collection. Thank you, Kirsten, for agreeing to be with us today. And our final panelist will be Peter Keach. Peter will bring a particularly important dimension to our discussion today from his vantage point as manager of engineered barrier science at the Nuclear Waste Management Organization. In this role, Peter is responsible for developing science-based descriptions of engineered barriers used to contain nuclear waste, and these in turn are used to gain an understanding, an increased understanding of possible degradation processes that could lead to leakage. And the approach is very comprehensive. It involves investigation of chemical, radiation, mechanical, and biological processes that could lead, in theory at least, to leakage from nuclear waste containers. And it's interesting in that among the possible corrosion degradation mechanisms that Peter and his team are somewhat preoccupied with are the potential effects of the microbiological community on these containers in these deep underground areas where they're stored. And so we look forward to hearing about that, Peter. Peter earned a PhD in electrochemistry from Queen's, sorry, I've got that wrong, from the University of Calgary, my apologies, and holds master's and bachelor's degrees in chemistry from the University of Guelph. He has authored more than 40 peer-reviewed publications and is the most recent chair of the International Workshop on Long-Term Prediction of Corrosion Damage in Nuclear Waste Systems. Peter, welcome to Waterloo. We're pleased to have you here to participate in this session. And indeed, I'd like to welcome all of our panelists, Alexis Dolphin, Andrew Doxey, Kirsten Mueller, and Peter Keach, to Research Talks, and thank them on your behalf for taking time out from their busy schedules to share their thoughts on this important topic with us today. As noted at the outset, and I'm going to say this one more time, each panelist will make a brief presentation. I'm just kidding. Feel free to use your 7 minutes. And we will then move into discussion mode. So Alexis, you're first up and the podium is yours.
So within anthropology, many of you know about the folks that work with living people. I work with the dead ones. And so this means that I work with human remains, usually bones and teeth. Part of my job is going to excavate at various sites, but a lot of my job is working with samples of mine and those of other people to look at teeth specifically for signs of what was going on in a person's environment during their childhood. Because teeth grow in utero and during the early years of childhood, and they permanently document things like stress, what people were eating, and exposure to pollutants. Even though they're thousands of years old, we're able to know what they were doing, a little bit about their health conditions, and exact periods in their lives where they were being affected. So part of this then in the past for me has been doing things like this, stable and trace element analyses of teeth and documenting when people were being weaned and all of this. But the point is that my goal in reconstructing environments is, gives me great information, but there's something else about teeth that's really cool. Well, part of it is that they preserve really well, but it's not just the teeth that preserve well, it's all the stuff on the teeth that preserves really well, or in the teeth. Some of you may have heard of various ancient DNA studies where researchers are using tooth dentin. So not the hard crown that you can see in your mouth, but the dentin that makes up most of your root, and inside the pulp cavity to try and get information about ancient people's DNA. But I'm actually not as interested in that as I am in this. So dental calculus is also a mineralized material. It's a mineralized biofilm, and it's the thing that your dentist picks off your teeth for you when you go. But not everybody has had that benefit over the years of having a person cleaning their teeth for them. And so across all human populations, and going back very far in time, some examples of, or remnants of calculus have been found on ancient hominid remains that are getting back as far as 9 million years ago. This preserved plaque. So this is just a picture of a human molar, a lower molar, that you can barely see the white little bit of crown poking out. This one is a really exceptional case. So it builds up. Sometimes you can section it and see incremental growth in this material. And so it mineralizes and preserves really well. And one of the cool things about it is you can find lots in it. It's kind of like coprolites. You love to find the poop and the plaque because it keeps a lot of good stuff in it. So it's got human DNA. It has microfossils, things like pollen. And then you can find activity waste, but then of course bacteria. And it's a fairly new area of research, but people are looking at doing things like identifying specific bacteria that indicate something about why humans have developed really bad problems with things like periodontal disease and caries. And then also trying to get to the point of reconstructing oral microbiomes for ancient populations and looking at how these populations of bacteria are changing as you see culture change happening, which is where I'm really interested. Like the origins of agriculture or the shift to industrialization and how that affects people's food and their environments. So this is a fairly new area that I'm only just stepping into. There are lots of questions still about sampling strategies and how to best get the DNA, and concerns always with things that are dug out of the ground with respect to preservation and contamination. So for me, the reason why I should explain why anthropology fits into the WCMR is just that we have access to direct evidence from people that goes back thousands of years and that provides a record of what they were eating and what they were doing and what they're exposed to and how over time they and their interactions with commensal bacteria, but also with pathogenic bacteria has adapted and modified with these big changes. So part of this here is that I had reached out at one point to Josh Neufeld, who I think I saw here somewhere. Yes, hi. And so he's been so kind to help me out with an honors thesis student to explore methods for trying to do some work with calculus here on campus. But long term, my goal is I have new funding for a 4-year project excavating remains from a site in Jordan with a history of 7,000 years of changing technology that has resulted in a polluted landscape. So it's the earliest part of the world where environmental pollution caused by humans from the beginning with a shift to advanced mining has, what is the impact on the people over time? And how can the calculus and what's inside it help us to explain that? Okay. I have no idea if that's 5 to 7 minutes, but I will leave it there, okay? All right, thank you so much. Okay, let me do this.
All right, thanks for having me today. I'll also try for 5 to 7 minutes, but I don't know if that's going to happen. Okay, so I'm a computational biologist, and so I'm going to tell you about the evolution of microbes from a bioinformatic and genomic perspective. So this is a genome. I know many of you have probably seen images like this. It's a circular bacterial genome, and now because of DNA sequencing, we have more and more of these. We have close to 184,000 microbial genomes that have now been sequenced and are available in the database. And this really allows us as bioinformaticians to ask some fundamental evolutionary questions. And so one of the key questions that we ask in my lab that I'm going to talk about today is, what do genomes tell us about the evolution of microbes? And so one of the ways that we've attempted to answer this question is through building a computational framework. And this is recent work out of my lab. So we've built this framework called ANOTree. And ANOTree, what it does is it takes a massive amount of sequencing and bioinformatic data and it combines it together into an evolutionary framework. So what we've done is we've taken over 26,000 species, constructed a tree of life out of that. Okay, so this is the most recent microbial tree of life based on all genomes. And what we do is we then combine that with the raw genome level information. Okay, so these are the genes and other genomic traits that you can see in the sequence information itself. And there's really an enormous amount of data. We have almost 155 million genomic traits that are included in this framework. And this framework is something that everyone can access. So we've got a nice web server which allows you, all of you I hope, to interactively explore this data and ask really fundamental evolutionary questions. So what the user can do is they can really start with any gene or any biological function that can be represented by a gene, and they can ask, where does this gene or function occur in the tree of life, and what is its evolutionary distribution, what's its evolutionary history. And so what our framework will do is really paint that trait onto the tree of life. You can visualize what lineages have it, what lineages don't have it, potentially where did that trait first evolve in the evolution of microbes. And so by taking all this information and looking at taxonomic distribution, this really gives the researcher an idea of the evolutionary history of really thousands of gene families and biological functions. And so just to give you some examples, suppose you're a researcher interested in the evolution of photosynthesis. If you query our framework with a photosynthetic gene here, you will see a region of the tree of life light up. That region contains matches to that photosynthetic gene. And so not surprisingly, you can trace that back to a specific lineage, that being the cyanobacteria, that's dated back to about 3.5 billion years ago. If we take a different trait, let's take an antibiotic resistance gene, something that is very problematic today, even if we map that trait onto the tree of life, we can see that it potentially has an ancient origin as well. And so this particular gene here appears to date back probably to the origin of the Firmicutes, and even that trait might be around 3 billion years old. But then if we take another trait, something like bacterial rhodopsin, okay, so this is a light-sensing protein, and we look at that evolutionary distribution in bacteria, we see that it's widely scattered. It's scattered across the tree of life. And this is very interesting because these cases like this are indicative of something called horizontal gene transfer. This are genes that are really jumping from species to species. And so one of the questions we've asked in my lab is, what's more common? Do we find genes behaving like this in microbes, or do we see patterns more like this? And of course, this has implications for understanding microbial evolution. And so we've asked this question for all known gene families, over 28,000 of them, and we find that overwhelmingly, most bacterial and most prokaryotic genes behave like this. So they're highly patchy, highly scattered on the tree. They're jumping around, and very few behave in this manner. And so we're very interested in these. We're interested in finding these genes that are mobile and jumping around bacteria. And I'm just going to tell you about one of these. And that is our genes that encode bacterial toxins. This is another area of work in my lab. These are 6 different toxins in their evolutionary distribution. These are all toxins that cause human disease. And what's really interesting is that they are quite dispersed. And in fact, they are far more abundant across the tree of life than researchers are aware of. And I'll just focus on one of these, and that's the Clostridial neurotoxin here. This is actually the most deadly toxin that's known. It's the most potent toxin known to science. And it's the causative agent of botulism and tetanus. And so what's interesting is if you zoom in on that case and you look at the tree of life, then you can see that that toxin occurs in this lineage here. This has been known for a long time. This is the lineage of Clostridium botulinum. But we can actually find this toxin. It has evolved, or it has been transferred to additional lineages across the tree of life. And our further analysis suggests it actually originated in Clostridium, and it's been transferred across the tree like this. So not to end the presentation here on a scary note, but some of these bacteria are actually quite concerning. So last year, we identified the emergence of this, or the transfer of this neurotoxin gene to a concerning pathogen. This is a hospital-acquired infectious disease pathogen called Enterococcus. And I'm showing you here the evolutionary tree of Enterococcus. Clade A is non-pathogenic. Clade B, these are all these tend to contain members of Enterococcus that are associated with hospital infections. And so we were able to predict and identify the emergence of this botulinum neurotoxin right in the middle of this tree here. And we know that it was transferred from Clostridium because when we look in the genome and we look at the arrangement of genes here within Enterococcus, there's an amazing degree of genomic similarity. So we even see the exact same gene arrangement when comparing it to Clostridium botulinum. So likely what has happened is Clostridium botulinum here has transferred its toxin to this emerging pathogen of humans, probably via a mobile element like a plasmid. Sorry to scare you. But you can imagine that this created some attention, so it's got a lot of press and scientific attention. The good news is that there have been no infections or outbreaks in humans due to this toxin yet, but it shows that we need to keep looking through the data to see the continual evolution of microbes. So I know this talk is about ancient microbial evolution, but my point is it's ongoing. So just to summarize here, we can use genomics, bioinformatics to infer these ancient events, but we can also see from the data that microbial evolution is very much continuing to this day. It's driven largely by gene transfers, and we see it happening a lot in hospital pathogens. And so with that, I'd just like to thank my lab here, this team that built Unearthing, and also my lab at UW. And I think I went over 7 minutes there.
Okay, so the first thing I like to do when I'm giving any kind of talk, and I'm probably going to go over 7 minutes because I'm trying to jam in like 20 years of research into like 5 to 7 minutes. But I like to try to convince people about why the red algae are interesting. Of course, I find them interesting, but why would you find them interesting? So this is a picture here that shows the various different kinds. Even this green one here is a red algae. These are commonly found out on our Pacific coast. They are found in fresh waters, and they are found around the world. So why the red algae? Well, Andrew just mentioned a second ago that photosynthesis evolved about 3.5 billion years ago. And that occurred in a group of what we call algae, or you can call them bacteria, or cyanobacteria, such as this, which is a picture of one of these up here. So we know that photosynthesis then ended up in some other groups. And one of the groups that it went into, and this is kind of a rough view of a phylogeny of organisms that have nuclear genomes. And what we have is a host cell that took up a cyanobacteria and then kept it inside for photosynthetic purposes. Why digest it when you can keep it and make it work for you and make you food? So here we have the green algae, which are right here. This is a really beautiful one. We have the red algae, and they fall within this group that we called plantae. And of course, you're probably familiar with the land plants. So why are the red algae important? Well, you'll see these dotted lines over here. This is another event that happened where you had a host cell that decided to take up a red algae and keep it as a chloroplast and make it do some work for it. And we ended up with some really important lineages, including the diatoms. Most of our food webs in freshwater and marine environments are dependent on diatoms. So leading from great whales all the way down leads to these organisms. We're probably going to see a bloom out in the Pacific within the next 2 months. The large kelps that we see out on the coastlines, both of these have chloroplasts derived from red algae. And then we have another weird group, lots in the news this past summer, red tides off the coast of Florida, which are dinoflagellates, which are in this group, where we had a host cell that took up a cell from one of these groups here that originally was from a red algae that originally was from a cyanobacteria. So kind of a complicated history when it comes to photosynthetic life, but the red algae have played a really important role in this. And kind of just to go back to teeth for a second, you all hopefully brushed your teeth today. Products from this organism here, Chondrus crispus, are used extensively as thickeners in food and toothpaste as well. So what do I study? Well, you heard the word Bangiales. Little do you know, but you all know this group. And I'm going to tell you why you know this group in a minute. So what we have here are these interesting kind of flat sheet organisms. This is called Porphyra, as well as a whole bunch of other new names that we actually presented, and we received a Paper of the Year award for it. You'll understand why in a second. And here's another one that is this little filamentous form. So we've got flat sheet, filament. They look really different, right? So you'd say one is one and one is the other. They look like apples and oranges, two totally different organisms. Well, it's a little bit kind of more complex than that. But before I go on, I just want to talk about this organism here again. So this is a picture from NASA's Landsat 8 satellite. And you can kind of just make out in here all this kind of hatching that you see in the water. This is South Korea. These are all seaweed farms. There's some oyster farms in here as well. And what are these farms farming? They are farming nori. Most of you probably know what nori is. Nori is the seaweed wrapping that you see in sushi. And so this is an incredibly important organism in terms of economic stability in some of those regions. So understanding how to identify species is really important. We're looking at $2 to $5 billion per year in economic output from some of these countries, including our own now. We do have extensive farms of this on the East Coast as well. So I want to continue on talking about the Bangiales being ancient. So Andrew mentioned 3.5 billion years for photosynthesis. We know somewhere in there that a host cell took up a chloroplast and made a red algae, made a green algae. But one of the interesting things about this group is we have some really fascinating fossils. One, Bangiomorpha pubescens, identified from the Proterozoic in the Canadian Arctic, dates back to 1.2 billion years. It is a filamentous form. It also had evidence of sexual reproduction, and that is because we could see structures that indicated to us that sexual reproduction was occurring. So that's really kind of neat. The other thing is it is identical to Bangia, that filamentous one that I showed you before. So we're talking about multicellular life and sexual reproduction occurring about 700 million years before the Cambrian explosion, before we started seeing a lot of those animals.
In 2017, yet another paper, again you can see this filamentous form, pushes the date of filamentous red algae back another 400 million years. So we're looking at a really incredibly ancient group. So in my research, a lot of the work I've looked at is trying to understand how do I identify species? What names do we give them? If you have a protected nori farm that has a certain species, but you can't identify it because a flat sheet it is really hard to identify, what do we kind of look at? Well, when we started doing some kind of gene analyses, what you see here is the pink ones are flat sheets. Some purple ones in here that are slightly different. Here are flat sheets, and then these green ones are all filaments. What are the question marks? They're ones I still have to name. I have to sit down and name, and I need to find time to sit down and actually name them. I've got a whole bunch of genera and a whole bunch of species that I need to identify. So just saying a flat sheet is a flat sheet and a filament's a filament is not as simple as that. They've been here for 1.6 billion years. They've switched back and forth between filaments and flat sheets. And so we need to kind of figure out what's happened in the evolution of this particular organism. And so what we've done now is we've actually sequenced the genome of Bangia atropurpurea. This is a freshwater invader into the Great Lakes. We've sampled it all around the world. It is nearly genetically identical. And the weird thing about this one is that it actually doesn't undergo sexual reproduction. All the other ones in this tree undergo sexual reproduction. But these ones, for example, don't. So our next question is looking at, okay, we know sexual reproduction happened a billion, 1.2 billion years ago, but what's happened in this particular lineage? Did they lose the genes for sexual reproduction, or are they having sexual reproduction when we're not watching? So our hypothesis was they've lost the ability to undergo sexual reproduction. We can't even get them to do this in the lab. And this is just kind of a life history. This is an asexual loop here, and then we have this sexual life history here. So we went into that genome that we have. We pulled out all the genes for sexual reproduction. There's a great paper that talks about the meiotic toolkit. If you have all these genes, you can undergo sexual reproduction. And guess what? They all have the genes for sexual reproduction. And the genes appear to be functional. So all of a sudden, we have an organism that's in fresh water, that's asexual, but we don't know what's really going on there. So some of the things that we're going to start looking at next is seeing if there's some way we can induce in a laboratory this kind of sexual reproduction and try to understand more in terms of the relatedness of different organisms and how sexual reproduction as well as multicellularity arose such a long time ago. So, of course, a lot of this work could not be done without my amazing graduate students, and I just want to note Vanessa Poletto-Borges, Trowena Shea, Mike Lynch, who's not pictured here, was a PhD student that finished a number of years ago, Saeed Amin, who did a master's project with me, and Mohini Nima, who was a grad student, but actually her undergraduate project was looking at this particular group. And thank you very much.
Okay, I'm going to talk quite a bit differently than the last few speakers because I'm really going to focus on some of the applied work that we do. We have a very, very good team of people working in this area in fundamental areas within it. And my job is to really try and consolidate a lot of the topics together, as we mentioned earlier. So I'll start off with a completely random looking slide of the Pickering power plant. The reason I'm putting that up there is to give you some context of where our problem begins at the Nuclear Waste Management Organization. About half of Ontario's power is made by nuclear, and then the fate of the waste, the spent nuclear fuel, is to go sit in these big ponds for 10 years or so, and then sit in these dry storage containers, which are very well shielded, and you can walk around or mostly stand around like these OPG people. But they can stay there for about 100 years. And then that's a very safe interim storage solution. But where it gets difficult, or where our job comes in at NWMO, is the long-term permanent solution. This is our graphic that we put in practically every presentation now. What it's trying to show is that eventually the waste will be put underground into a facility about 500 meters down in host rock. It's got to be the right kind of rock, and we're looking at very ancient rocks in our siting process. So both sedimentary, which are hundreds of millions of years old, or the Canadian Shield, which is in the billions. And so those things have impacts, in terms of the microbial programs that we run, in terms of the age, as well as some of the other subcomponents. Here's a close-up of what we call the repository room. And so we have used fuel container, and I can give a very long talk about that goes way longer than 7 minutes. But there's the fuel loaded in there and then put into clay barriers. This is highlighted here because this is where a huge amount of our microbial work is. The clays that we use are bentonite clays, and the formations are typically a couple 100 million years old. And so they carry with them some native microbial species, which then impact our work in terms of material degradation. So our design is really based on natural barriers like the rock and these engineered barriers as are shown here. Okay. What's really, really important to us is microbially induced corrosion. Here's my chemistry slide. I am, in theory, a chemist, although not today. That's the reaction just to show that sulfide can be formed by microbes underground or in other places, basically in anoxic areas. And once you perturb an underground, often it'll go through a sulfide producing cycle until eventually it goes to methanogenesis. But for us, this is the really problematic one, because this sulfide species can react with our material here. So this is an experiment that was done by the Swedish program, and you can see the black stuff is a corrosion product. And if that happens in the repository, then you're going to have problems, because that's where you could, in theory, have a leakage of your container. So it's really a corrosion issue that we have, that it forms the basis of why we're doing all the microbial work that we're doing. Really, and this is very important, our engineering design has a huge component that's based on the microbiology. So this is a plot. Again, this one comes up a lot. Basically, it's showing, so this is the density of the clay. So we make these clay features here into bricks or big blocks, or we pack it in really tight. Because if you get it in very, very tight, then you can prevent microbes from growing. So this is basically a microbe activity. And in the green zone here, you're at the background levels. So if you pack your clay densely enough, then you can suppress the microbial activity in the vicinity of the used fuel container. So in this case, the microbial processes are driving the engineering design. As I mentioned, we have a very integrated program, and this is a little graphic to show some of that. We have recently, our researchers at universities have recently been awarded an Ontario Research Fund grant. It's multi-million, multi-year, 9 faculty members, 5 universities, including right here at Waterloo with Josh, as he's already been flagged before. But all more importantly, the actual work is done by Katya, so made sure I put her up there as well. What we're doing is we're trying to, as I said, a lot of applied work, and this is the work that team is involved in. We take, this is just a schematic. We drill down into underground labs, host rocks that are suitable or comparable. They're typically in Switzerland because they're very good at the underground work. And we stick these things, as you can see, these are little container materials and the bentonite clay. And then we expose them to real conditions so that the microbes that are in the groundwater fluids or in the host rock can do their thing. And then we can see if our designs are working. So we have high densities and low densities to show when we have microbial activity and when we don't. And typically the container materials are carbon, steel, and copper. So I'm not going to get into the processes behind that, but that's typical for most of the waste organizations, is to look at those materials. And so this is a multi-waste organization program. This is just a few slides just to show some of this stuff. That's what the bentonite clay looks like when it's in its pellet form. We also have little blocks that we do. And basically we load the coupons in and shove them into anoxic water to saturate them. And then we ship them in these things. and then they go off into the underground labs. And so these are pretty busy slides. I'll highlight this one here because this is where we do water sampling. So we pour water, run it through, so we can do microbial analysis on that. And then we expose them, and then we chop them all up into the clay bricks, which we do a lot of microbial stuff. We take swipes of the coupons and so on. We're doing it all anoxically in the glove box there. That's me standing around mostly, but I'm also keeping something out of the way of the guys that are actually doing the work. But as we do the emplacement and retrieval of our modules. And these are the very oversimplification. I'm sure when I get to Josh's slide, he'll just cover his eyes or something, because I'm making it so simple. The point here, though, is when you have the low densities, these are the densities here of the actual Montmorillonite, and I won't get into that, just why they're different than those ones. But when you're at the low density of Montmorillonite, you can get more activity, and then the high densities do suppress it. So that's what we're looking for. That works for both anaerobic and aerobic. And then this is Josh's one. You probably cover your eyes, Josh, because I've really dumbed this down. But what you can see is the ones that we're worried about that are the SRBs over here, they're only present on the outside of the container. And these are the things here in the borehole fluid in there. But as you move inward, you go through other ones that are a little bit more rugged, I suppose, on the outside of the container to the very outside of the bentonite. But right at the core, you're into these things that are spore formers, the relics, the stuff that you may find in the bentonite anyway. And those are not likely to be harmful to the container. So this is the first of many analysis that we're doing on these types of processes. And how are we continuing on this? And where it gets really interesting from us from the design and working with our site assessment teams and so on, we want to do these even deeper. So those are just 30 feet deep in the underground labs. We're looking, this is one of our boreholes that we've developed up at one of our siting locations. It's actually 1 kilometer long, and we want to put these things at 500 meter depths. So we packer them off above and below, let the groundwater come in slowly, do some verification of that. So before we have an underground lab of our own, we're doing some work on the site. And so again, we'll be pulling these modules out periodically. That's just the core. There's endless meters of core coming out, and that's just sitting around. We don't know what to do with it right now. And another analog that we're exploring is ocean. Very active microbial community outside. And we're trying to, these are some of the things, we've only done one of these at a relatively shallow depth of 100 meters up in Saanich Inlet on the west coast. So this has just been in, we've only done one to do some method development. Eventually we'll go into very deep ones so that we can combine the effects of pressure, because eventually if there's a glacier over top of a repository, you can get to tremendous pressures. So again, this is the brand new work that we're really just getting underway. This is an engineering slide, so incredibly boring. But the important thing here is this yellow zone here. That's where almost all of our corrosion allowance is. So the wall thinning that happens is almost exclusively because of the microbial effects. So somewhere out far away, there'll be sulfide coming in from the microbiology. So this becomes site and design specific. So again, depending on the repository, where it is, if it's sedimentary or crystalline, what we're getting out of this, we'll have to refine this allowance. And so this is a big part of our work on the engineered barrier science team. And so part of that, moving into the damage function, why we need all those other people as part of that ORF. We want to get a 3D image of it. So if we have just a uniform sulfide, it turns out that it's quite specific in our emplacement room where the damage will be. So this is just multi-physics, groundwater modeling, right on top of that microbial understanding. So when we look at those things all come together, we really do need an integrated approach to come up with our overall damage that we might expect in a nuclear waste container. So here's my summary slide, hopefully 7 minutes after I started. Again, we're looking really from materials degradation. We have tons of fundamental work, which I didn't cover today. And what we find within that work is we're really pushing the frontiers of things like low biomass, very hostile environments for microbes because it's dry bentonite, very dense. So we're working in very challenging things to come up with an understanding. And then the thing I haven't really covered is we're looking backward to in time for our sites and so on. But we also have to model very far forward. 1 million years of a safety case. So it becomes a very, very difficult analysis to try and come up with. The last thing is, this is going to be work that we're going to keep doing for many years. It's going to roll into future R&D programs, and all the waste organizations are interested in this kind of work.
Thank you, Peter. Thank you very much indeed. No, not yet. And indeed, thanks to all of the panelists for your presentations, which I think will set the stage very nicely indeed for the best part of the event today, which is the discussion with the audience. So I wonder if I could ask the panelists to take the chairs at the front of the room, please, where you can face the audience and they can see you. You know, as I listened to your presentations, I couldn't help but think that that microbes are both, well, they're good microbes and they're bad microbes, and they present a real paradox to humanity. And the two concerns. One, of course, is leakage of nuclear waste. That's a big concern. And the other is all these mutations that are taking place in hospitals that are giving rise to all kinds of toxins that are going to cause us grief. So maybe during the discussion we can focus on the good things that bacteria do. All right, so the way this is going to work is as follows. I need to present to you some procedure details. This session is being recorded, and that means that in order to feature in the recording, when you speak, when you ask a question, when you make a comment, you need a microphone. And so, yes, Rebecca is going to be the bearer of the microphone to you. Just raise your hand and she'll make sure that you have a microphone. And also to the panelists, when you speak, please speak. I don't think that was caused by a microorganism. Please speak into the microphone. All right, so the floor is now open for questions and comments from the audience. And you can direct your question or your comment to the entire group of panelists, or you can direct it to a panelist or two panelists, whatever you wish.
Hi, just wanted to say first, it's a lot of fun hearing everyone's talk. These are all really interesting. It's great to get a little bit of a surface level impression of them. I have questions for everyone, but I'll start with just one for Dr. Dolphin. Pronouncing that right? All right. So something I've heard about using teeth for any sort of reconstruction is that certain models don't work after, say, the 1950s due to atomic testing just because of the amount of radioactive material thrown into the air. And you mentioned 7,000 years of range in studying Jordan and the shifts in the over time as technologies are developed and advanced. And I'm wondering, are there any other sudden developments that obfuscate particular markers or things that you would normally use, say, in a hunter-gatherer population to identify this, that, or the other, say, diet, deficiencies, changes in the atmosphere, that become obfuscated because of other technologies that have been developed over time?
Let me see if this is working. That's a great question, and it's interesting because the consensus seems to be that if we're not, that the, if you're thinking strontium-90, I would assume, perhaps, that is not something that's considered as having any sort of impact on our ability to look at trace elements or stable isotopes and how they behaved in ecosystems in the past, or how we can interpret what they mean. Everything that I'm familiar with is assumed that you have a sort of a uniform ability to apply these techniques going back thousands and thousands of years. So it's a great question. You know, you got me thinking a little bit, to be honest. But yeah, I mean, people are using these and assuming they work a certain way, and that because of that, they can look for changes associated with culture change, but not really where it's making it where we have to maybe probe the data a little bit more. But I think, thank you. I think you're making the job harder, perhaps, but I appreciate it. Thank you. I don't know if that's much of an answer for you, but.
Yeah, thanks everyone for fascinating talks. I guess my question sort of Andrew and Peter maybe together. So it was striking that Andrew was looking back using his technique to follow the sort of evolution or transfer of certain traits across the microbes that you're looking at, and Peter's interested in projecting forward, I gather, along the same lines, how do you do that? Like, can you use the information that you've gained from looking backwards to try and make projections forward as to what's going to happen to certain traits?
I guess I'll start. So that's a very interesting question, and there has been a lot out there in the literature even about whether evolution is predictable. Certainly it is going back in time, because we can start with extant data, organisms that live today. We can look at their genomes and we can reconstruct backwards. Looking forwards, I would say some things are predictable if we know the kind of selective pressures that are going to exist within an environment. We might predict what type of genes or functions will be advantageous and therefore increase in frequency. So I think some adaptations going forward will be predictable. So maybe looking at nuclear waste, the radioactivity and decay, that might impose a certain selective pressure on those environments. So maybe certain genes will get will be more beneficial than others, but in general, I think it's actually very hard to predict in most cases.
Thanks for that. All right. I guess that question is, I mean, that's a real worry for people, especially on engineering teams that don't understand any of this stuff. The strategy we have is we don't look to predict the evolution. What we try and do is we try and make the environment as hostile as possible and make the issue happen as far away as possible from the waste container. Now, we do have some things that work in our favor, in that we have radiation that comes and it certainly helps to suppress microbes nearby. We put the clay in very dry and very dense, and then there's a heat, there's a thermal bloom that really dries it out. So those things help us to have confidence that if these things are going to happen, they'll tend to happen farther away. And then the material selection is very important too. The copper itself is relatively good as an antimicrobial substance. The bentonite, if it when it starts to get wet as water comes in, it will swell and really help squeeze out microbes. So those are thoughts that go into the engineering design along the way. But for things of this would probably fall into the unknown unknowns, or the general uncertainty that comes with designing any system that's going to last a very long time. And so we flip over to rely on our safety assessment to see the consequences of something like this happening. So if the containers fail, what are the consequences for radionuclide transfer, or how will the species that are harmful flow out of the containers? And we try to mitigate that by making it a diffusion-only system. So the rock is very large and intact. That's part of the reason it's deep. The clay itself, as I mentioned, it swells. It's self-sealing. So that helps encase everything into a system that will make everything happen very slowly. So by our engineering design, we work as much as we can. And then on the other side, like the stuff that I work on, we work with the R&D community to try and be on the very front edge of any of these things. And we're going to continue that R&D all through the process of site investigations, excavations, all of the processes right through the point where the site eventually stops being monitored by a decision of a future generation. So those are the strategies we have. But I mean, anyone that has other strategies, we're always listening for those things as well.
I think there was another hand up there, yes. And you've got the microphone, even better.
Thank you. So I had a question directed towards Professor Dolphin and Professor Doxy concerning gene transfer and the movement of animals. So for a gene transfer to occur, I assume there needs to be some physical proximity between the two microbes. So could you use that to glean the geographical history of microbes, as well as the animals that perhaps may carry these microbes, such as the migratory patterns of humans, and trying to date microbes in one area, get the age of that microbe in a particular area to predict when the humans moved to that area from another geographical area?
Okay,
I'll start with the first question about can you infer maybe a shared environment when you observe gene transfer? I think yes, you can, right? Especially if the closer the genes are together, let's suppose you see one version of it in one microbe, another in a very, say, distantly related microbe. If those are very similar, you can use kind of molecular dating and infer that those there must be some kind of recent transfer. And the only, I think, conceivable way is if they at least share some kind of shared environment, yes. About inferring kind of migration patterns of humans based on that, I'm not so sure about that. That's a very interesting question, though.
I'm not going to be of much more help there. I'm not really sure.
I can comment on one of the things that I didn't touch on in my tree is that a lot of the biogeographic patterns in the distribution of organisms do show up in phylogenetic trees. And often when you have an organism that is appearing in a part of a tree, grouping with a bunch of organisms that are in that locality, but it's from another location, you can actually try to pinpoint how it's moved. So the seaweed that I study, as well as many others, have been transported not just by humans, but also by all sorts of marine life. And you can pick up those patterns of transportation around the globe, and you can infer generally over time how quickly they've moved. And you could probably tie that into horizontal transfer as well if you're seeing these genes appear in certain organisms that maybe you wouldn't expect, and then mapping that into larger phylogenies that have that biogeographic trend.
Okay, thank you, Kirsten. Other comments or questions? Yes, over here, please. Rebecca? We'll try and get it synchronized so you can
the best ways to
focus on one side of the room at a time.
Hi. Oops. Okay, that's a good distance. I'm just wondering, when you're taking into account like the evolutionary history of all microbial life, I always thought that it'd be very important to keep track of like how many generations these microbes are undergoing, and to track like how mutable they are. Because when we say something's like 3 billion years old, like what are we talking about in terms of generations? Because I've heard that in a lot of these environments that they live in, unlike the lab, they're very nutrient depleted, and so their actual life cycles might be very longer than we would typically expect. You know, not like E. coli, which can like has a doubling time of like 20 minutes or something like that. So I'm wondering if there's any way to intuit what the natural doubling time is of a lot of these organisms, and how we take that into account when we consider evolution.
Maybe I'll start and then I'll pass it over to you. You're right. There are highly variable rates of evolution between microbes and at a genomic level as well. So you can see that in the data. You can see mutation rates being completely different from lineage to lineage. And so we do actually take that into account mathematically in the models that reconstruct these phylogenies. So yeah, you can actually end up finding the best, most likely model that accounts for what we call rate variation. So yes, it's built into the model, and you're right, it's that it is complex, and we probably oversimplify things too.
Okay, very good.
Regarding the 3.5 billion organisms, so those are cyanobacteria, and those are fossil stromatolites. And so we have good fossil evidence that support the origin of some of these organisms, similar to what I talked about with the red algae. The stromatolites today, which are mats of cyanobacteria interspersed with sediment, we see living ones like those as well in Australia. When we're trying to probe groups that have really ancient evolutionary histories, one of the key things is picking out genes that can help you differentiate between those different organisms where there hasn't been so much evolution happening that you can't get a phylogenetic signal. So the genes that I typically use, which are the small subunit for the ribosomal gene, if I sequenced all of mammals, I would see very little difference between all of mammals. animals. But within the red algae, because they're so ancient, we can use that gene to kind of probe those evolutionary questions. If we start getting into generation time, yeah, we're not kind of working on that. We're trying to look at those broader type questions. But the genes we look at, such as ribosomal genes, are not prone to high rates of evolution because they have such functional importance.
Okay, very good. Bernie.
So this question, I guess, is mainly directed at Andrew and Kirsten. Do you have a way, I guess, of getting a sense of maybe groups of microorganisms that might be more or less susceptible to gene transfer and kind of separating that from selective pressure? Is that hard to do? I'm just wondering, like physically, are some of them, you know, say refractory to having gene transfer? Since you've surveyed on such a big scale now.
So yes, actually that was a question that we asked of the data. We kind of searched the tree and looked for what regions of the tree are undergoing the most gene transfer. And certainly there are regions that light up. You know, so there are many to talk about. I would say, so the human gut microbiome, for example, is one environment where gene transfer happens regularly within the Enterobacteria within the human gut. So because you have this common environment and there's lots of gene flow, you can see that in the data. So I think, yeah, the question is maybe more of environments, right? Do some environments drive gene transfer? And a lot of that is through mobile elements like plasmids, right? So certainly, and that might even change over time, right? If we go back in time, maybe we see different environments where there are higher rates of gene transfer. So yeah, it's a fascinating question.
About 15 years ago, I published a paper that looked at the horizontal transfer of a mobile group 1 intron, and it actually had jumped from a fungus into a red algae. And more recently, what we've discovered with a lot of these ancient organisms is that they have a lot of symbiotic relationships with bacteria, with fungi that have probably persisted for millennia or hundreds of millennia. And in the process of sequencing the nori genome, we tried to get this organism as clean as possible and kill every bit of bacteria and fungus on it, and we actually ended up killing the red algae. Right, yeah. So we've had a lot of struggles when it comes to that. So we're still early in the genomic era of algae. You know, we're kind of behind. Lots of animals are done. But we're starting to learn a lot more about possible gene transfer between these microbes, even into eukaryotes. We do know they're really dependent on each other. So I think those are environments where you can see that happening as well.
So just as a follow-up, do you have a sense overall, is gene transfer speeding up? Is it slowing down? And I'm sure it's different for different groups of microbes, but...
Yeah, that's a good question. Yeah, so I'd say in general, when you kind of paint gene transfer onto the tree, the one sort of major pattern that you see is that it is happening quite frequently in the tips of the tree, meaning in the taxa that are sort of existing, I would say today or somewhat recently, right? So it's going back in time, it's actually quite hard to predict because these genes jump around so much. So how can you statistically tell when it's happened in the past? So, but we do see it happening quite broadly across the tree in recent lineages.
So it's still a challenge then to get a sense of whether it's accelerating or decelerating overall. Yep.
Okay, so we have a question here, but I wonder if I could also ask a question at this juncture because it follows on nicely from Bernie's question, and then we'll get to you. Is that okay? So I'm going to direct this question in the first instance to it actually has application to all of you. When I look at your microbial tree of life, I see data. And I'm also struck by the fact that genomes are changing because of gene transfer. And so my question really is, to what extent could we use artificial intelligence to predict from your large cache of data such things as the likelihood of gene transfer, such things as detecting genes that have the potential to become toxins, etc. And again, I'd like to hear from Andrew, but from all of you, because I think this, wherever you have data, you have in this day and age opportunity for using AI.
So that's an excellent question. The reason I'm smiling is you just mentioned a future, well, I would say current research project in my lab with the toxin prediction. So we are starting to use AI to try to predict these toxins. And I think you're completely right that AI can identify patterns in this data that humans can't. Regarding the tree, so one thing I just want to clarify, it's probably more Laura Hugg's tree than our tree. We based a lot of it on Laura's work, and yes, again, I think AI, using it to understand the patterns in the tree is a great idea, particularly when it comes to grouping genes by function. So as humans, we like to take these genes and put them into categories based on our understanding of biology. If you use AI may see these categories from the data in a much better, more efficient, or more correct, I should say, way than we can. So I think that's one really great use of AI, is in actually trying to categorize these genes into function and understanding what functions seem to be very mobile on the tree. Yeah.
Okay. So, Peter, could you comment on this? Perhaps from the perspective of being able to predict what microorganisms are going to be a problem when it comes to safety of storage, maybe using some of the information you're getting from your colleague at Waterloo?
Sure. I guess one of the issues that we have in general is when you start to say, well, microbes aren't a problem, then the microbial people raise their hand and say, here are 10 problems, each of them. And so, you could have, in our case, we have radiation, we have heat, we have all of these things, and there are bacteria that can survive all of these, right? If we look at the survivability of bacteria, or archaea, if we really want to get down there, we could have an issue if these things, coalesce on individual microbes. We're trying, in general, to prevent that by our design. But if this sort of tool does develop, we would be right among the first to jump on it to see if there are ways that we could take this, these predictions of how these things could come together, and then iterate that through our design to improve our design, and then back. And I could see a really iterative process that could follow from that, whereby we keep enhancing our design to try and prevent, one of these things from taking off.
Anyone else want to comment? Okay, so let's move on to this question here. Can we have a microphone over here?
Sorry, I didn't see who it was. Thank you. Oh, Carly.
Hello, yes, thanks for an excellent talk. My question is about there can only be one. So it seems like one I'm familiar with is that in history, photosynthetic apparatus was so difficult to evolve, it kind of only happened once, and then everybody kind of stole it. So is there a similar thing about sex? Would you say that sex only happened once and everybody copied it? And then if I switch gears and ask Alexis, I'm ignorant about a lot of anthropology, and I'm wondering if in human history, if there's examples of technologies or something that only happened once and then everybody had to copy it.
So I'll address, photosynthesis. So right now we're still not sure as to whether or not there was one event or two events in terms of the origin of photosynthesis. So we have the lineage that is the greens, and then of course they just shared, and we have the lineage of the reds. So our kind of working hypothesis is there's probably one event that kind of led to photosynthesis. When it comes to sexuality, considering that we have this meiotic toolkit that you need to have all these genes, and of course being able to undergo sexual reproduction is really advantageous in terms of the evolution of organisms, because if you can have offspring that are slightly different from you that then have the possibility of surviving, I'm leaning towards more that there was likely this meiotic toolkit in the beginning, and then it was shared somehow amongst these host cells. But we are so far back in time here that we're really kind of not clear what some of these host cells would have looked like that had taken up things for chloroplasts. But I think that's kind of beyond my area of research. I've always kind of focused more on kind of what's happened in my groups. But in terms of the evolution of chloroplasts, I think, and the evolution of photosynthesis, I think we're understanding that a little bit more. But the other area, yeah, that's a great question. I think I need to look more at that.
Okay.
Yeah, with respect to the technological question, I guess I would just say that if there's one thing humans do that we talk a lot about in anthropology is they have lots of sex with each other and they share their ideas all the time. Maybe at the same time. I'm not sure. So I'm just saying this is what my courses are like. So I guess what I'm just saying with respect to technological advances, humans have been sharing ideas and moving around and interacting with each other really since before we'd even necessarily call ourselves humans. So it's always messy with us. Okay, that's my answer for everything. Okay.
So could I just comment or ask a question to Kirsten, I guess, really? The question is, are chloroplasts, as we know them, more efficient at photosynthesis than the red algae from which they originated? And the reason I ask that is because there must have been some evolutionary change within the photosynthetic apparatus once it got incorporated into a host cell.
Yes, so when you look at the different lineages that have chloroplasts from either red algae, so we would call those secondary events, and we also see that in the green algae, there often tends to be a reduction in the number of membranes down because there needs to be some sort of nuclear control over the chloroplast. When we look at plants, the small subunit of the rubisco gene within green algae and plants has been transferred to the nucleus. But when we look at the red algae, that never happened. So the red algae actually seemed to retain more of its photosynthetic genes than the green algae in the plant lineage. So in terms of efficiency, you know, I'm not sure I can answer if one's more efficient than the other. The red algae and the groups that come off the red algae have accessory pigments that make them able to capture light at really low light levels. And so that makes them able to occupy habitats such as, you know, 500 meters down in the marine environment where you might not expect photosynthetic life.
Okay, so once again, we need artificial intelligence to sort this out.
Yes, that would be good.
But, given the power of evolution that we've all experienced, I can't help but think that the chloroplast in a modern-day plant is much more efficient at photosynthesis than the original red algae. That's what I would predict.
Possibly,
absent
yes.
artificial intelligence. Ladies and gentlemen, we have come to the magic hour at the other end of the spectrum. I'm told by Rebecca that we are supposed to close at 1, and it's shortly after 1 now. I would like to ask you to join me in thanking our panelists for a very, very exciting session. So if we could do that now, please.